Hello, listeners. Yamina here. Welcome to this episode of the Dr. GPCR podcast. Before we jump into this episode, I'd like to share a few things with you. The December and last Dr. GPCR newsletter of 2020 is now available. If you haven't subscribed yet, please visit drgpcr.com/newsletter. Also, don't forget to share the newsletter with your colleagues. We cannot wait to bring you brand new Dr. GPCR newsletter editions in 2021. Second, we will be taking a short break of the Dr. GPCR podcast in January 2021 as our family will be welcoming a baby boy. But rest assured, we are already working on bringing you brand new episodes, starting sometimes in February 2021 with an amazing lineup of guests. Also, stay tuned for a major announcement regarding the podcast. We're also proud to announce that we are pursuing consulting opportunities in the GPCR field. For help with your R&D project, please visit drgpcr.com/consulting or reach out by email at hello@drgpcr.com. And last but not least, we will be closing this first season of the Dr. GPCR podcast with a series of interviews with phenomenal female scientists in the field. We hope you'll enjoy it. Good morning or good afternoon, wherever you are, listeners. I'm Dr. Yamina Bershish, founder of Dr. GPCR, and this is another episode of the Dr. GPCR podcast. Today, I have the pleasure of having with me Dr. Jennifer Pluznick. She is an assistant professor of physiology at Johns Hopkins. Hi, Jen. How are you?
I'm good. How are you?
Good, thank you. It's so great to have you today with us.
Yeah, thanks for having me.
Absolutely. Excited to chat today about a very interesting set of topics, might I add. So you're now an associate professor. What led you to where you are today? Could you tell us a little bit more about your career trajectory?
Sure. Well, I wanted to be a scientist since the time that I was in elementary school, but I had no idea what that meant, really. So as a first-gen college student, I just knew that I liked figuring out new things and I liked learning about science, but I wasn't sure that I wanted to be a medical doctor. So I thought being a scientist sounded really cool. As an undergraduate, I think I started to figure out that that I really did love science, but that the types of jobs that I thought were really fun were the jobs that I saw the PIs doing, where they got to decide what new thing to explore next. And I slowly learned that maybe I couldn't easily do those types of jobs with a bachelor's degree. So that was basically the basis of my decision to go to grad school. So in graduate school, I became fascinated first by the kidney. And then during my postdoc, I became fascinated both by the kidney and by the GPCRs. And I'm still fascinated and working on both of those things today.
That's super. Was there a moment while you were an undergraduate when you said to yourself, well, or maybe you'd taken a course that introduced you to GPCRs, or was it during your postdoc when you got introduced to the GPCR world?
It was really during my postdoc that I got interested in the GPCRs. I do remember learning about orphan receptors in graduate school and being really fascinated by the whole concept of orphan receptors. But my research didn't go in that direction until I was a postdoctoral fellow.
Wow. We all have a memory in our trajectory where we got first introduced to GPCRs, and then we thought they were fascinating machines, and then the whole signaling cascade, and some went on studying signaling, some went on studying specific receptors, but we all do share that commonality, definitely. So you mentioned that you're working on the kidney and GPCRs in the kidney. Can you tell us a little bit more about the work that you do?
Sure. So the focus of our lab is really to understand the role of what we think of as understudied GPCRs and kidney function. So by understudied GPCRs, I mean GPCRs that are understudied in the context of the kidney. They're not necessarily understudied overall. So we think we're especially fascinated by things like olfactory receptors and taste receptors and orphan receptors, which we know are expressed in the kidney, but which have been really unstudied or pretty severely understudied. So there are some really classic famous GPCRs like the angiotensin II receptor, arginine vasopressin receptor that have been studied in a lot of detail in the kidney. But there are these other receptors, some of which are expressed to similar levels if you just look at transcript levels, but which haven't been studied at all. So we're convinced that if these receptors are studied or expressed in the kidney, that they're likely to play important roles, or there's some reason that nature is expressing them there, and we want to understand what they're doing. And I just think it's really fun to take a receptor where you have no clue what it might be mediating. You don't, in many cases when we start these projects, we don't know what the ligand for the receptor is. We don't know what the cell type of expression for the receptor is, which sounds kind of crazy and risky, and sometimes it is. But if you can go from that to figuring out what the ligand is and where it's expressed and what it's modulating in terms of kidney function, I just find that really intellectually fun.
Wow, it's so funny because when you think of taste receptors or smell receptors, you think they're all on the tongue, for example, or in the mouth and in the nose, and that stops there. But turns out they're everywhere, and it's understudied and underappreciated, I think, at this point. What is? Do you have a favorite receptor among these chemoreceptors that you're studying in the kidney?
So, although our lab is studied a lot of different orphan receptors and other GPCRs, I think I first got into this area because when I was a postdoc, I kind of accidentally found olfactory receptors in the kidney. And I think olfactory receptors as a class will always be my first love in terms of GPCRs. So as a postdoctoral fellow, I was just doing a microarray comparing some control and diseased kidney cells models, and on that microarray we saw that olfactory receptors were some of the top hits, and I was really confused by that at the time for the reasons that you just stated. I thought, well, that olfactory receptors are found in the nose, this is a kidney cell line. I famously said that I wasn't sure that I could really trust the data because these crazy receptors came up as the top hits. But my postdoc advisor, who's much wiser than I, said, like, olfactory receptors in the kidney, though, like that could be really cool. And somehow when he said it, it sounded like a much better idea. And so I really got fascinated with the idea that olfactory receptors are really just chemical sensors. And if you think of them as chemosensors, then I think the idea starts to make a lot more sense. So I think my favorite GPCRs would have to be olfactory receptors. The other thing I really like about studying ORs or olfactory receptors in the kidney is that it's a concept that I think the general public can also understand and can kind of get it. I can talk to my parents or my relatives who aren't at all scientists, and they can grasp the concept that you have these chemical sensors in your nose, but it can also be used in other places in the body, and they can kind of appreciate that it's cool. And I love getting able to talk with family about that in a way that they can understand, which I think is pretty unique for the type of science that I've done in the past. It can sometimes be really hard to explain some of these concepts in a way that is approachable, but I feel like the idea of smell receptors playing roles in other places is something that other people can kind of understand is cool.
They can definitely relate to. When I usually talk to non-scientists about GPCRs, I usually say that they're expressed everywhere in the body, and then you can see, smell, and taste thanks to GPCRs, and people very easily can relate to that.
Exactly.
Speaking of olfactory receptors in the kidney, what is the status of research? What do they do in the kidney, if we know?
Yeah, so, well, we're starting to know. So we've been slowly trying to study the individual receptors that we've been looking at. So one of the receptors that we've studied seems to play a role in blood pressure regulation by regulating the release of a hormone called renin. That's a key step in the main blood pressure regulation pathway, the renin-angiotensin pathway. Another receptor that we have studied in the past is a receptor that plays a role in how the kidney handles glucose. So those are two cool examples because those are both really obviously physiologically relevant processes. So obviously blood pressure regulation is a huge issue when it comes to hypertension. It turns out that the other receptor that regulates glucose actually does so by regulating a pair of transporters, one of which is a drug target for type 2 diabetes. By modulating glucose loss in the urine, you can lower plasma glucose levels. So we've been really lucky with some of the receptors that we've started, where we literally started knowing absolutely nothing about the receptor. We didn't know the ligand, we didn't know the localization. And it turns out they're doing things that I think you can make a case are physiologically or pathophysiologically relevant. That being said, there's still a huge hurdles to overcome for those receptors as well as for a lot of the other ones. So a common issue with olfactory receptors is that we don't always know what the ligands are. So some of them are still orphan receptors. For the ones that do have identified ligands, they often have multiple ones. And so then figuring out which of those are the physiologically relevant ones, which of these are going to be circulating in a kidney, or which of these are going to matter to a kidney cell is a kind of a different level of deorphanization. There's also a lot of questions about functional orthologs when you go between mice and humans, different model systems. So I think that there's still some huge hurdles that we need to overcome in order to understand them much better.
Wow. So how many of these receptors have you identified in the kidney that seem to play a role in a physiological event or events?
So so that's a great question. I have to think in my head. I think that our current count of olfactory receptors in the kidney is in the teens. But there's only those two, are the only two that we've studied in some detail. There's some other orphan receptors that we've also studied in some detail that play roles in different parts of the kidney nephron as well. So if you count those orphan receptors, then I think maybe there's 5 receptors that we've studied in a little bit more detail. So the orphan receptors the receptors I tend to think of orphan receptors are the GPCRs whose nomenclature starts with GPR, which means that they were an orphan when they were originally named. So for example, we've published on GPR 41 and GPR C5C. So we've found some of those GPRs are actually expressed at pretty high levels in the kidney transcriptionally, especially compared to the ORs. They're found at much higher levels. So we've started looking at some of those as well.
Wow. And what are the tools that you usually use to identify these orphan receptors and to study them functionally?
So the tools that we wish that we had were good antibodies, but as most GPCR people probably already know, we don't always have good antibodies, and in fact, we normally don't have good antibodies. So in order to, for us, we think about these receptors, there's 2 big questions we try to answer when we first begin to study a receptor. One is, where is it found in the kidney? We hope that if we know the cell type, because the different cells in the kidney have such specialized jobs, we hope that if we know the cell type, that'll give us a clue. And then the other question we always ask is, what does this receptor sense? What is the ligand for the receptor? So those drive the tools that we're looking for, right? So for localization, if we can get an antibody, that's wonderful, but we often can't. So we use reporter genes. So we have mice where we'll have the gene of interest is knocked out, but the original promoter is driving a reporter that we can use. We also use RNA scope. We've used microdissection followed by PCR. We'll use whatever tricks we can to try to get at where the receptor is. The other big question is, what is the receptor sensing? And for that, we really want to know what agonists the receptor has, and ideally antagonists too, but we can rarely get quite to that stage yet. For that, we have a number of different assays in the lab, cyclic AMP based mostly, where we can run them through screening assays and try to find new ligands, confirm published ligands, and get a sense for what might be a physiologically relevant activator of the receptor in the context of the kidney.
So in the context of these receptors, most of them are GS-coupled receptors. Are they from a class A, or how are these categorized?
Yeah, it's a great question. So the olfactory receptors are we think of them as GS coupled, and the nose are actually coupled to G-olf, which is an olfactory G protein that's basically the olfactory version of GS. But it increases cyclic AMP. It does the same type of thing. But those receptors seem to have no problem coupling to GS as well. I think G-olf and GS are similar enough that at least in an in vitro situation, they can translate pretty well. The orphan GPRs are a mix, obviously. So some of them are GI and some of them coupled to all kinds of different things. So those are a little bit trickier, but the olfactory receptors are certainly all increasing cyclic AMP. And I think I forgot the second part of your question.
How are these categorized, whether they're class A or...
Right, gotcha. Yeah, so I think most of them are class A receptors. The olfactory receptors are further broken down into a couple different different types of olfactory receptors, and most of the ones that we've been studying are the class 1 ORs, but there are certainly other classes that are also found in the kidney.
I never, until I looked up your profile, I didn't even stop to think about the fact that there's olfactory receptors in the kidney that actually have an important physiological role. Speaking of physiological roles, if they're important physiologically, there must be attached to some kind of a disease. Can you tell us a little bit more about the disease areas that these GPCRs are involved in the context of the kidneys?
Yeah, so far each GPCR that we've studied seems to do something completely different. It doesn't seem like they're all GPCR sensors, but it doesn't seem like they play roles in only one area. So the kidney is a regulator of homeostasis for the whole body. So I mentioned earlier the example of the one OR that plays a role in blood pressure regulation, another one plays a role in renal glucose handling. The GPRs that we've studied, one of them also modulates blood pressure regulation. One of them modulates acid-base balance. So it seems like each of them is helping the kidney to kind of fine-tune its ability to maintain homeostasis under various, different insults that get thrown at it. But we haven't noticed any particular theme in what types of things they do. It seems like of all the millions of things that the kidney can do, the GPCR will stand in to help fine-tune various different processes.
And in disease states, you had mentioned, I think, when you were talking about your postdoc research, that you were looking at gene expression in healthy tissues and in also disease state tissues. What were the cells that you were looking at, and what were the differences in olfactory receptor expression?
Yeah, so in that initial experiment, we were looking at a cell model of a disease called polycystic kidney disease, which is actually the most common inherited disease in the U.S. Like 1 in 500 to 1 in 1,000 people have that condition. You're born with completely normal kidneys, but over time your kidneys become overrun by cysts, and so people go into renal failure, but typically not until mid to late adulthood. And when we initially compared those, we saw differences in olfactory receptor expression between them. When we went back and repeated those experiments, the differences in olfactory receptors weren't always consistent. So we always had ORs on the top of our list, but they weren't always the same ORs. And I've never quite figured out what to do with that, honestly. I'm not sure if it's because those were microarrays and the ORs, as your listeners may know, are all really similar to one another. And it's very possible that we had some RNA that was cross-hybridizing to more than one spot on the array, and that's what was causing those issues. We've never gone back and looked at that in a more careful way, and we really probably should. But we've instead just focused on kind of following each OR and asking it what it does, and then trying to figure out the physiological or the pathophysiological relevance from there. So we're certainly interested in the receptors that our lab is focused on now in terms of hypertension. So we've been looking at some hypertension models. We've also gotten interested in them in terms of heart disease because we see some changes in cardiac parameters as well. Some of these receptors are also expressed throughout the vasculature, not only in the renal vasculature. And then certainly in diabetes, obesity, for the receptor that plays a role in glucose handling.
Yeah, and if the more you understand about their physiological role and how they function, maybe they could be used as new drug targets or in a combinational fashion, using them with current drugs that are already existent in the clinic.
Yeah, absolutely. I mean, that's one nice thing about studying GPCRs, right, is you always have that hope that if you have good agonists or antagonists that you might be able to modulate this pathway in a more straightforward way.
Definitely. So you mentioned that to study these receptors, what you would hope to get as soon as possible, or as much as possible, are antibodies to try and detect the expression of these receptors. What other tools or initiatives do you think could help speed up targeting or identifying these orphan receptors or these olfactory receptors in the kidney that would help understand their biology?
Yeah, so antibodies is a totally correct answer. I think good agonists and antagonists are another thing that we need for a lot of these receptors. And kind of related to that, a weird problem about olfactory receptors, which has gotten better, but still probably isn't as good as it should be, is the fact that these receptors, when you express them exogenously, so if you transfect them into HEC cells or COS cells or oocytes or whatever model system you use, they tend to get stuck in the ER, and they don't get to the cell surface. So you can imagine that for most ways that we screen receptors for ligands, that's a huge problem. So if your receptor is not on the cell surface, it doesn't matter how cool your screening system is, you're never going to get a hit. So another thing that that's gotten better, people have come up with various tricks to help these receptors traffic much better. But there are still some receptors where we struggle to get great cell surface expression, which definitely makes it hard to even do the agonist or antagonist screens that we want to do. So I think that's a weird little cell biology quirky problem that needs to be overcome even a little bit more solidly for us to really be able to de-orphanize the entire class. And then the other thing I would say is a lot of these, I kind of alluded before to the fact that identifying orthologs between different species is difficult. So I think that's another pretty big problem, actually. So there's for olfactory receptors, there's about 350 of them in human, and there's about 1,000 of them in mouse. So you can imagine that when you try to draw a straight line between which one belongs to which one in the other species, it gets pretty complicated. If you blast a human sequence against the mouse genome, it might match several different mouse receptors somewhat equally. So which one is the ortholog? Is there an ortholog? So one approach that our lab has been trying to take is trying to find what we think of as functional orthologs. So an ortholog that has a really similar ligand profile. So if the ligand profile of the potential ortholog matches that of the human one, then we're more convinced that it might truly be the ortholog. Many times in our hands, the ortholog that's listed in PubMed, when we test agonists, the responses are very different between the human and the mouse receptor, and then we wonder if they're really functionally orthologous or not. So I think that's another issue. In order for this research to be translatable, is we really need to understand which of the mouse receptors have a human ortholog, so that we can be thinking about that translation in the proper way.
Wow, I didn't know that there were issues expressing these receptors in a transient way, for example in HEC cells, and getting them to the plasma membrane. Could you talk to us a little bit more about these, and what are the tools that people have developed to get more of these receptors expressed so that you can enable a screening, for example?
Yeah, sure. So I think the most famous and the most helpful tool was developed by Hiro Matsunami's lab at Duke. So they found RTP1S, which is a chaperone protein that you can co-express with the receptors. And for at least a subset or a cohort of the ORs, it makes a huge difference. We have a number of ORs in our lab that won't go to the cell surface at all if we express them on their own. But if we co-express them with RTP1S, we do see cell surface expression. Another tool that our lab has developed that several other labs are now using as well, often in combination with RTP1S, is something that we termed the Lucy tag. So this is a leucine-rich tag that naturally occurred on another protein that was trafficked very well to the cell surface. So I had a really smart graduate student, Niranjana Natarajan, who during her rotation in my lab came up with this idea that maybe if we put this leucine-rich tag on the front of an olfactory receptor, it would help it get to the cell surface. And it turns out that she was completely right. The really cool thing that I like about this tag is that it's a signal sequence, so it's cleaved during processing of the protein, so it's not on the final protein that gets to the cell surface. So you're not burdening the protein that you're studying with another tag, which we always worry could alter ligands sensitivities and such. So we use the Lucy tag a lot. That's really helpful. Another tag that's used a lot in the field is a Rho tag, which is the part of the N-terminus of the rhodopsin protein, which is also just something that's trafficked much better than olfactory receptors are. So there is a whole cadre of tools. We often use the Lucy tag and the Rho tag and RTP1S, and sometimes we still have issues with surface expression. Or we can get it to the surface, but it kind of limps there. And so if you don't get great ligand responses, you wonder if it's because your surface expression is poor, or whether it's really because you're not testing the right ligand. It's a really interesting problem because in the nose, obviously these receptors traffic just fine, right? Like they don't have any problem getting to the cell surface. So it's something, it's some weird issue of exogenous expression that I certainly don't yet understand. But there are a lot of different tools that you can throw at it that make the problem better, but don't always completely solve it for every OR on the table.
Wow, the Lucy tag approach, I think it's a really neat approach, because since you mentioned it gets cleaved off and it's just, you get the quote unquote clean receptor at the cell surface. It's really a neat technique.
Yeah.
Do you know if in the nose, the cells, do we know if there's any other chaperones that we should be maybe looking at to help this? Or is there a way to study olfactory receptors in a more, I want to say, more physiological context? Or is there a cell line that we could be using that's closer to the native cells that they're expressed in?
Yeah, that's a great question. There are chaperones other than RTP1S that are found in the nose. In our hands, adding those into the hex cells doesn't usually solve the problem. You can certainly, we've also tried, and I'm sure others have tried, transfecting the ORs into olfactory sensory neuron cell lines. We haven't had much luck with that approach. You certainly can study them in situ, so to speak. So you can have a load olfactory epithelium dissociated from a mouse with calcium and flow ligands after it. One approach is then, if you see a cell that responds, you then stick a pipette and sequence it to find out which OR was expressed in that cell. Or you can have some sort of GFP reporter so that you know which OR which of the 1,000 ORs you're looking for responses from. But obviously those are technically a lot more difficult than plating hex cells. Because we're really interested in, so I think that those approaches are amazing and are used by a lot of labs that are interested in these receptors when it comes to the chemical sense of smell. But because we're really just interested in the renal side of things more, we tend to use the hex cells as a very imperfect model system to try to get a sense of the ligands. But yeah, I think that there are, if you really just want to know about the ligand sensitivities of the receptor and you're willing to look at it in a natural olfactory epithelium context, doesn't make for great high throughput screening of 1,000 different receptors, but it does allow you to get maybe a better idea of what it's really being activated by in vitro, since you don't have to worry about cell surface expression issues.
Yeah, hex cells are really cool and easy to use, but I think everybody knows that. Everybody who uses hex cells knows that they're easy to use, easy to plate. As you mentioned, they grow like weeds, and you can just dispose of them. And they also give you a good idea on what happens with a GPCR. But on the other side of the coin, there is also a need to study these receptors, any receptors for that matter, in a more physiological context, because that actually matters. Still,
absolutely, yeah.
How neat these olfactory receptors are. And I think the fact that they're unable to go to the plasma membrane where they're transfected in hex cells might also show that there is a very tight regulatory mechanism in their native tissue, which still I think, and correct me if I'm wrong, is poorly understood, or not as well as understood as we'd like to have it.
Yeah, I think your instincts are exactly right. So in the nose, there are in the mouse, there's 1,000 different olfactory receptors, where there's something called the one cell, one receptor rule, where each olfactory sensory neuron will pick one and only one of those olfactory receptors, and it will express the heck out of it, and it will suppress the expression of every other receptor. And that's how smell works. It's the labeled line signaling. So if one particular neuron fires, your brain knows that that had to be olfactory receptor 19 because that's the only one expressed there. And there, I have heard thoughts in the field that maybe that ability to suppress 999 other receptors, maybe something about that is what it makes it hard to just randomly express it in another tissue. Maybe there is some sort of, and maybe that's why we can't just transfect it into an olfactory sensory neuron, because that olfactory sensory neuron might have all kinds of ways to prevent the spurious expression of an extra receptor, which will completely ruin the label line scheme. So yeah, I think the fact that they have to be under somewhat tight control might have a lot to do with the fact that I can't always express it where I want to.
Still, I know it's hard to work with these receptors because you can't express it, you don't have antibodies, and you need more tools, but still, I think they're really cool. It's a really cool system that needs more work, obviously, which is great in many ways. Do you know of any olfactory receptors that are currently investigated as drug targets, or do you know what kind of drug targets would olfactory receptors be, if any?
Yeah, I don't know. I don't know of any olfactory receptors that are currently being investigated as drug targets, but that could just be beyond my knowledge. I don't think that necessarily means there's not any. I think that olfactory receptors could certainly be good drug targets. Some of them are expressed. There's a couple that are expressed beyond the nose that are expressed in lots of different tissues, and those might not be great drug targets because they're expressed in a lot of places. But there's some other ones that seem to be very specifically expressed in very specific tissues, and those you can imagine would be much potentially better drug targets because you could have a more targeted effect of your compound. So I think as long as you have that tissue-specific expression that, they're like any GPCR, they're on the cell surface, they should be accessible to a compound. If you have that tissue-specific localization, then I think that there's a lot of promise there.
I agree. I absolutely agree with that. Oftentimes, when it comes to using GPCRs as drug targets, there are a lot of key information that might be missing. One of them would be more the pathophysiological pathways that are involved with these GPCRs. In the case of olfactory receptors, or in the case of GPCRs in the kidney, let me rephrase that to that. What is the key information that you think is missing to help us, target these receptors in the kidney when it comes to diabetes? You had mentioned hypertension. Can you tell us more a little bit about that? What's your opinion about it?
Yeah, so I think that that for a lot of these receptors, the ones that our lab has studied, we know something about where the receptor is found under control conditions, and we know something about sex differences of expression under control conditions. But experiments that that we really want to do and that we just need funding and time to do are to look at how that expression changes in different disease states. So I think if you're going to use it if you're going to try to activate or block that receptor as some sort of therapeutic, then you can't just assume that the expression in disease is the same as it is under control conditions. And I think for a lot of other genes, we know that that's not true, that they change quite a bit, especially if they're involved in the pathways that, for example, if a protein is involved in pathways that regulate blood pressure, I think it's naive to assume that there's going to be no change in expression in hypertension. And that would include not only the level of expression, but potentially the cell type of expression, right? Which could completely change how a therapeutic might interact with it. So one thing that we're eager to do is to try to look at the expression of these receptors in much broader contexts. So we've looked at males and females, for example, but what about pregnancy versus menopause? What about, during aging? What about let alone in different disease states? So I think that's something that that's really missing from the field, just because the field is kind of young, that needs to be looked at over time. Even if the answer is that it's static and that there's not a lot of change, I think that's a really useful answer, and that we need to know that information.
It is important. The way I look at it is like having this map where you can look at different cells in a normal tissue versus a disease state, and you can kind of have this list or this interconnection between the different partners, different GPCRs, different signaling pathways, and try to identify what changes between the two states. And that can be helpful. Even if it says nothing changes for this particular pathway, there must be some molecular basis to the disease.
Yeah, that makes a lot of sense.
So you were saying that the field is young. That gets me to my next question. What is your advice to junior scientists who want to work on olfactory receptors or GPCRs and contribute to getting those maps set up and get that knowledge become common?
Yeah. So that's another great question. So I think one thing I would say is that you can learn from others in the field. That's definitely an amazing thing to do, but I wouldn't be held back by the opinions of others in the field. I think that sometimes, I think that always we need new brains in any field that don't know the way that we know. Of course it works in such and so a way, in order to find new things, in order to realize that maybe it doesn't work in that way. So my example would be that everyone kind of knew that olfactory receptors were not expressed outside the nose until Hans Hott's group and others started to report that actually they were found outside the nose and that they might do important things. So I think you need to follow your data, even when it surprises you, even when it might go against what you assumed to be true before you started the experiment. I think that's how you can get lucky and stumble on new and kind of cool things.
Kind of like Mr. White in Breaking Bad when he was saying that the chemistry must be respected. I think here the biology and the signaling must be respected.
Absolutely.
In that sense, you had talked to us a little bit in the beginning about the expression pattern where you discovered during your postdoc that there were olfactory receptors where there shouldn't be. I want to ask you a little bit more about what I'd call the aha moments that you had as a scientist in your career. Were there any moments where you made some discoveries and you said, well, this is now shaping my opinion on such and such?
Yeah, so I think that the one you mentioned was one of them. So when I first found that those ORs came up on the microarray, I really wasn't sure what to make out of it. But an experiment that my postdoc advisor and I came up with was to order, although there's not antibodies for the thousands of different mouse ORs, and we didn't really know which one to look for, there are antibodies for the G protein and the adenylate cyclase that are responsible for smell in the nose. So the olfactory specific G olfactory protein, and the adenylate cyclase 3 is the olfactory adenylate cyclase protein. So an aha moment I remember from my postdoc is we got those antibodies and I put them on a kidney and I double stained, and those two antibodies localized with one another, and they localized with one another to one of the only cell types in the kidney that was thought to be kind of chemosensory in nature. And when I saw that localization under the microscope, I remember thinking like, okay, this is my new project. Like, this is really cool. This is something that I definitely want to follow up on. Another aha moment I can think of from my postdoc is one of the olfactory receptors that we study in the kidney. When we're trying to understand the ligands for that receptor, it turns out that it's activated by chemical compounds that are made by the gut microbes. So these are things that circulate in your blood at low millimolar levels, or pretty high levels, but they're generated by your gut microbiota, which means that the activation of this receptor is somehow tied to the metabolism of your gut microbes. This is a receptor that modulates blood pressure, so that means that your blood pressure regulation is somehow tied to the activity of your gut microbes. And that was something that that still kind of blows my mind, and is something that we're still kind of trying to figure out.
I think so too. I think the fact that you have a microbiome that regulates so many events in the body is still a fascinating field, and it's something that we definitely need to look into more. And it gets us to the fact that nutrition is important, exercise is important, and it helps us understand why those components are so important.
Yeah.
You mentioned just minutes ago when we were talking about young scientists and what was your advice to them, and you were mentioning that having people coming from different environments when they don't know that in the field this is how it's done, are very helpful. For example, your student who came up with the leucine tag to go to help express the receptors of the cell surface. What are some ways that you can think of that we could increase diversity in the field, and obviously would be beneficial to studying these receptors?
Yeah, so I think that increasing diversity in the field is important, and you guys gave a couple great examples of why having diverse viewpoints and diverse mindsets can be really useful. When I think of increasing diversity in the field, I think especially of URM scientists or LGBTQ scientists or women scientists. And one thing that I think is important is that increasing diversity in those ways needs to be seen as a responsibility of all scientists. I feel like in the past, there's been a sense that promoting women in science is a job of other women, or that promoting URM scientists is a job of other URM scientists. And I think to really affect change, we need to realize that all of us have a responsibility to fix these inequalities and to try to move towards making things better. I think there's a lot of different ways that we can do that. I think, if you're inviting speakers at your university, or if you're on a program committee inviting speakers, you can be really conscious of how diverse those invitees are and speak up if they're not diverse, if you're in a position to do that. I think if most universities have some sort of programs to try to promote diversity in science, and I think that, in science, I feel like time is a pretty valuable commodity. It's kind of the equivalent of putting your money where your mouth is. And so if you think that these things are important, I think one thing you can do is to volunteer to help to mentor the students in those programs, or to go over the applications, or to be on the committee that goes through those applications or helps to run the program. So I think that we all have a responsibility to try to make science more diverse, in large part because that makes science better, and that time speaks loudly. It's none of us have a lot of time, but we all find ways to make time for the things that are most important.
Agreed. And you touched on two very important points there. What I want to add to that is, I think, promoting science at a young age to kids. You mentioned that you wanted to be a scientist very early on, and having the ability to interact with scientists as a child, or as a high school student, or as a middle school child, that's also very important. And the other thing that I want to mention is also mentorship. I think it is scientists' job to mentor their students, or mentor students, not necessarily the people who work in their lab, but also help them grow as scientists, and tell them, as your postdoc mentor had told you, no, I think this is cool. You found something that's really important, and that's what kind of pushed you into this direction, which is phenomenal to have.
Yeah, I think that great mentorship can go a long way, and can help to kind of iron over a lot of other issues. I've also, like anyone, I've had negative experiences with more senior scientists too, but one or two mentors who say like, hey, I think you can do this, really helps to iron over so many other issues. So having somebody who's in your corner, who you can go to if you have a problem, you can go to when you feel like you're not being supported by someone else, and get support from, I think that's really huge.
Exactly. And just being able to bounce off ideas, even if you feel like they're not necessarily the best ideas, but having someone in your corner who can easily tell you, well, listen, I'm not sure it's a good idea, and you take it,
right?
You don't take it personal. It gets you to think more. That's also very important.
Yeah, I think having someone where you trust them enough so that if they tell you it's not a good idea, they have your best interest 100% at heart, right? And they're not saying that because they want to steal your idea. They're saying that 100% because they're not sure it's a great idea. I think having someone you can trust on that level is pretty huge.
Agreed. We couldn't agree more on this one. Thank you, Jen, for being here today. I had a very great time learning more about OR receptors. Before I leave you, I looked up your website, and I thought you had a really cool lab website. And I also,
Thanks.
thanks. And I also saw that on your website you did have a link that said job openings. So if you have job openings, do you advertise them on your website only, or is there any other spot where you put on these job ads?
Yeah. So we usually do advertise them on our lab website. We don't have any right at this moment, but I always encourage people just to email me and check in and see if they think they might be a good fit for the lab, what we might have coming up.
That's great. That's great. And what I wanted to also tell you and tell the listeners today is that on drgpcr.com, we do have a career page. So if you ever have any open positions,
perfect.
Feel free to email us. The idea is to gather as many GPCR related jobs, whether it's a postdoc, whether it's an industry job, or whether it's just an internship, just to have them at the same spot so that people can go there and find their best GPCR match in that sense.
That sounds awesome.
Super. So the website is drgpcr.com/career. Anytime you can reach out, we'll be happy to post the ad.
Great. Thanks, Yamina. I really enjoyed this conversation. It was really fun.
All right. Thank you so much. Me too, Jen. Take care now. Bye.
Okay, thanks. Bye.
Thank you for listening to this Dr. GPCR podcast episode. We hope you enjoyed it. Subscribe to the podcast wherever you listen to podcasts. I'd like to thank our guest, Attila Forrest, Jin Chong, and Shivani Sachdev. Music by Rosa Bershish. I'm your host, Dr. Yamina Bershish. We're always excited to hear from you. Visit us at drgpcr.com or send us a note at hello@drgpcr.com. I also wanted to wish you happy holidays and a very happy new year. I'd like to thank this year's guests and thank you for being such a wonderful audience. Thank you again for the privilege of your time, and until next time, stay safe.